Good morning, everyone. I'm going to talk about more of the technical details, kind of an overview of the science, and some of the pros and cons of using CMS, how the protoplast fusion is done, and that sort of thing. So, one of the basic things that we need to start out with is how inheritance works in plants. And what I'm trying to show on this slide is we have two parents, male and female. These are individual cells here to talk about the transfer of the various genomes that are in plants. There are three organelles that encode for the genetic inheritance. One of these is the nucleus, and that's generally what we often think about when we talk about making crosses and selection and so forth. But there's also genetic code in the chloroplast and the mitochondria. And the important thing to our discussion here is that those are transmitted in different ways. You know, the nucleus goes through meiosis. There's a reduction in the number of chromosomes, and then it's reconstituted back to the parental level in the progeny. The transfer of the chloroplasts and the mitochondria is unilateral, only goes through the female line. Now, there are a few exceptions. Occasionally, these plastids will make it through down the pollen tube, but that's a very, very rare event. So this is typically what happens. So this is the basis for cytoplasmic male sterility, and what makes it so valuable is this maternal transmission. Okay, and usually, I think in every case that's known, it's a defect in a gene on a mitochondria. There's part of the machinery actually for the chloroplast and mitochondria is encoded in the nuclear genome, and if there's an incompatibility between the two, then that can lead to this type of sterility. As I mentioned before, the maternal transmission is of those is pretty much a given. And then the pollen is non-functional, or the anthers may be completely lacking. It can vary all the way from just inviable pollen to no anthers at all. But the ovules, the female side, is fertile and will receive pollen from a male fertile individuals. And this is a mechanism that enforces outcrossing. So that's why it has occurred in nature. Here's, for example, a picture of this is an arugula flower, and you can see the anthers here, and the style and stigma would be the transfer of pollen onto those that would allow self-pollination here, although there are other factors that would probably prevent it in an arugula plant. And then here's a picture showing a cytoplasm male sterility. If you look closely, you can see that there are no anthers here in this. This is another Brassica, this Brassica oleracea. So this is one thing. We could figure out which varieties are CMS, even if companies won't tell us, because you can grow them out, let them flower, and then look for this morphological difference, and that would tell you. So it does occur naturally in nature. People have found populations of wild radish and wild beets that have these has this cytoplasmic male sterility, and it's thought in those populations it enforces outcrossing, and it's part of the natural process. Another way that you can get cytoplasmic male sterility is through wide crosses. And for example, in beans, if we've found cytoplasmic male sterility by crossing scarlet runner bean with common bean. The two genomes are different enough, or the organelle genome is different enough from the nuclear genome, that you get this incompatibility, and it leads to CMS. It's kind of a hit and miss thing where you have to make crosses and look at what you're getting out of it, and some things will work and some won't in terms of producing CMS. So that's called alloplasmic induction of CMS. And then the other method for creating CMS is using protoplast fusion, a laboratory type technique. Now, I'll get to that in a little bit, but just to talk a little bit about the mechanics of how CMS is used to produce hybrid seed. You have to maintain those CMS lines, and you can't self-pollinate them to be able to do that. So you have to always be crossing them to a maintainer line, which is your male parent here. This is the female. Line A is the female. S stands for male sterile cytoplasm. N is normal cytoplasm. These other genes are called restorer genes, which are encoded in the nuclear DNA, and I'll get to those in a little bit. I'll explain why they're important in a minute. But this is the process you go through. You always take your maintainer line, which you have to maintain as a normal inbred through selfing, and then cross it to the female. The seed you'll harvest will all be CMS, and so you cross it again, and so forth. And that's just kind of a cyclical process. Now, when you're producing an F1 hybrid, you're using the CMS line, the line A, but you cross it to a different parent. It's going to have enough differences. You'll choose a parent that will give you some hybrid vigor and some other traits that perhaps you want in there. Basically just a cross, and then you harvest the seed, and that'll be the seed you sell as your F1 hybrid. Now, this is where the restorers enter in. If a seed crop is what you're after, say, for example, you're using CMS to produce canola, and the seed is what you want to harvest, then you need the restorer gene in order for that plant to be able to fertilize itself or fertilize its neighbors and produce that seed. And so you have to find your inbred with these restore genes. You cross these to the CMS line, and it's going to be heterozygous in your F1 hybrid, but that will mean that the plant is fertile and can self-pollinate itself. Now, if you don't care about the product you get, say, for example, broccoli or cauliflower, where the economically important part is produced before there's any seed, and that's what you harvest, then you don't need to worry about restorers. And so it would just be a cross between this and this. They'd be a recessive on the restorers. So that's the major difference in how CMS is used in different crops. This image gives you kind of a diagram of the protoplast fusion technique. Basically, you're taking plant cells of various forms, treating them with enzymes like pectinases and cellulases, which the pectinases allow the cells to separate from one another, and then the cellulases strip off the cell wall. And you're left with just a very thin membrane, delicate membrane around the contents of that cell. And those are called protoplasts. And so those are, if you're trying to do a protoplast fusion, you mix those together in a solution. You subject them to some kind of osmotic shock, like polyethylene glycol is often used in protoplast fusion, or electrofusion. We put an electric current through the liquid. And what happens is those protoplasts that are in contact with one another, those membranes will sometimes pop and then seal together, and you then get two cells that become one. Now, and these are called heterokaryons once they've done this. So they'll have double the chromosome number, or they'll have the chromosome numbers of each parent in the protoplast. Sometimes those nuclei will fuse, sometimes they don't. And then you have a mix of the organelles, the chloroplasts and mitochondria in those. Here are some pictures of protoplasts and then regeneration of the cells of various stages. These are our leaf protoplasts. You can see the chloroplasts in there very easily. You don't see the mitochondria. They're very small, and particularly under a light microscope, they're very difficult to see. These are what would be called suspension culture protoplasts. You take callus tissue and you grow it in a liquid suspension culture, generally without, they're generally not green, so the plastids are there, but there's, you don't find fully developed chloroplasts, so there's no green color. This is a picture of a petunia, actually a fusion product right here. You can make protoplasts from various tissues of the plant. In this case, they were made from petal tissue. And that red color in there is the anthocyanins in the vacuole in that petal. So it's kind of interesting. And it was fused then, I forget what the other species was here in this particular image, but it was fused with a mesophyll protoplast. So you get this, you can select the hybrids based on the fact that you have the chloroplast present and then that pigment present in the cell. Once you have these, you take these fusion products, you plate them out on various tissue culture media. They begin to divide. They form a cell wall again, and they begin dividing. And so you see something like this, early stages of dividing. And this is a hybrid in that it has both the chloroplasts here and then these other undeveloped plastids present in it. You can plate it out on a solid media, and in this case, we have little somatic embryos beginning to form some of these cells. They have the program to form a new plant, and so they're starting to do that. And then here's a callus tissue with somatic embryos that are coming out of it. So then from this stage, you would take those, move them into, well, you would have to put roots on them, then move them into soil culture, and then out into the regular environment. So in creating a CMS, or transferring the CMS, you have to do kind of a variation on the protoplast fusion. And the products out of this are called Cybrids. And what we have here is this is going to be the cytoplasm donor, and this is going to be the nuclear donor, because what you want to do is you want to put this nucleus into this cytoplasm. So you would treat these with something like gamma or X-ray irradiation, which would generally knock out the chloroplast or the nuclear genome. Then treat these with a compound like iodoacetate. It's a metabolic inhibitor, and it shuts down the cell. So these become non-functional. I should have X'd out the chloroplasts here. Those would also be non-functional. But then when you do your fusion, what you end up with is this nucleus then in that cytoplasm. And that would, you would hope that would give you the CMS. Generally, you regenerate a number of different events out of this, and then you select and you grow out the plants, and you see which ones are cytoplasmic male sterilla. Seems to be hung up on this one here. Let's see. Okay, yeah, I think maybe the animation was hanging up or something. Anyway, okay, now I'm going to talk about Brassicas specifically here, because this is the one where the controversy really is surrounded. So the Brassicas are a rather promiscuous group. And I think this slide kind of conveys it. And it's very interesting in evolutionary terms, too. There are 3 diploid species, Brassica rapa, Brassica nigra, and Brassica oleracea. I've drawn in the, what kind of crops, foods we eat that come from each of these groups. And then there are naturally occurring interspecific hybrids between these that have formed. Like the Brassica johnsia group is a hybrid between nigra and rapa. Brassica carinata is between nigra and oleracea, and then Brassica napus, canola and things like that, included in this group, is between oleracea and rapa. So some of the cytoplasmic male sterility has come out of things like rapa and put into these various other groups. Radish is included here because it's also been a source of cytoplasmic male sterility, and the main source right now. The lines here represent how easy or difficult it is to make natural crosses between these groups. Like rapa and oleracea cross fairly easily. Rapa and napus do, but it's more difficult to cross napus with oleracea. A cross like this can be done with brute force. You may be making thousands and thousands of hand crosses and eventually get some seed out of that. The upshot of this is it's possible to transfer some of these, make some of these cytoplasmic male sterols by using natural crossing. We haven't always had to go to CMS or to protoplast fusion to be able to do this. There are some wrinkles to this that I'll get to in a little bit here. So here's a list of transfers that have been done to produce CMS in the Brassicas, and then the method that was used. So you can see that there were interspecific crosses used with the rapa, with nigra. Here's a napus interspecific cross. The Ogura CMS is the main form of CMS that's used commercially right now. This came out of radish originally, and it was made both as through a natural cross, but then embryo rescue was used as well, another tissue culture type technique, which has its own controversial issues as well. We were successful in doing this, but what people found out was that these, yes, they were male sterile, cytoplasmic male sterile, but they had a cold temperature induced chlorosis associated with the chloroplasts that were present. The chloroplasts that came out of radish and were put into cabbage caused this particular problem. So then this is where the protoplast fusion came in to replace those radish chloroplasts back with the Brassica oleracea chloroplasts. And then we achieved plants that were cytoplasmic male sterile that did not have that cold-induced chlorosis. So it's, why did these other ones that have been made, why don't we have those as sources of CMS? I think there are various issues that one encounters with these abnormalities. The plants may not be as fertile. Here, for example, this is all in broccoli. This is a male fertile flowers, and if you look closely, you can see the stigma and the anthers here. Here's that cytoplasmic male sterile picture I showed you earlier, and then here are a couple examples of ones with flowers that are less normal. This one almost doesn't produce any petals at all, and tends to be fairly female sterile as well. So in some of these combinations, you get a lot of sterility, and it just doesn't work. For whatever reason, Ogura has been fairly successful. So can we produce natural CMS? This is one possible idea to how to get around this particular issue. You can make crosses, natural crosses, and obtain seed. Crossing directly from radish, yes, you could do that, but then there's this cold temperature chlorosis problem. which I don't see any way to get around without doing a protoplast fusion. There's other species. There's CMS that I've worked with called a non-CMS that comes out of Brassica tunaforte, and that one's that has its transfer has been done through protoplast fusion, but it's in theory possible to transfer it using natural crossing as well. There's a question about embryo rescue. Right now, I think tissue culture techniques in general are allowed in the NOP. They're not an excluded method, but will this continue? It really, if you talk about the integrity of the plant and some of the principles that Edith Lamberts Van Buren has laid out, then this would be a technique that should not be allowed. The bottom line is that economically useful forms of CMS are hard to achieve. So what sort of alternatives are there? There are things like sporophytic incompatibility, which some companies have used and continue to use. I won't go into the pluses and minuses of that right now, but that's certainly a possibility. And so another question is, how much does the organic community currently rely on CMS? It's mainly an issue in the cold crops, brassica cabbage and so forth. CMS is used in other crops, but for example, in carrot and onion, but that was naturally occurring, so it does not fit in this definition of excluded methods. I think chicory would be another one to add to this list as well, of ones where protoplast fusion has been used. There's another question here. Are there other traits that might have been transferred into plants that we don't know about? We tend to have short memories. For example, in tomatoes, the embryo rescue has been used to transfer various traits in from wild species. And like the tomato mosaic virus resistance we use in tomatoes, that was very widely used in many of the modern cultivars, but that came originally out of an embryo rescue type project. And so I think it's something that's going to confront the organic community, not just with this particular example, but with other traits as well. And I will stop there, I think, with what I have to say.